Polymorphic DNA can be detected in the absence of sequence information. कि हमें किसी DNA के sequence की information नहीं होती. Even then, we can map the DNA. For example, here we can use the technique which is called as PCR in order to amplify the particular genomic DNA, and in first instance we cannot use STS sequences. Like STS sequences are sequence tagged sites. They are mainly used to map the DNA, but here if such sequences like STS are not even known, we can amplify the DNA segment. And here, if you remember, we can develop arbitrary primer. I hope कि आपको याद होगा rapid की technique, that is random amplified polymorphic DNA. हम arbitrary primer develop करते हैं जिनका जो sequence length है it may be around 10 nucleotide base pairs, और इसमें जो GC content manage किया जाता है it may ranges from 50 to 80%. Another constraints that we can take into consideration while developing primer for rapid is that there should be no palindromic sequences in the primer. Palindrome means if the sequence they are reading the same, that is in 5' to 3' direction they are the same. Like 5' to 3' they may be adenine, they may be guanine, and so on. And same is for adenine, thymine, etc. So they should not be read same in 5' to 3' डायरेक्शन. जैसे मैंने कहा कि अगर कोई चीज़ लिखी हुई है ABCD है तो दूसरी 5' से वो ABCD की तरह उसके सीक्वेंस ना हो. It will prevent the self complementation between the primers. Rapid technique is very valuable for the mapping of genome of plants because it provides wide range of DNA markers so that we can know the location of different genes. But there are still certain limitations of the use of rapid technique, although it is valuable in the absence of sequence information. The limitation is that reaction mixture can influence the result. कि template concentration कितनी है, दूसरा solutions कितने हैं, एक ग्रुप के रिजल्ट और आएंगे, किसी और रिसर्च ग्रुप के रिजल्ट वेरिएबल हो सकते हैं. एंड इन द सेम वे एक और जो लिमिटेशन है कि हेट्रोजाइगोट्स जो हैं उनको हम डिटेक्ट नहीं कर सकते, कि जो दो अगर पेरेंट्स हैं तो उनके जो हेट्रोजाइगोट बनते हैं दे आर डिफिकल्ट टू डिटेक्ट. Because of these limitations, a modification has been developed where we can use STS sequences. कि अब हमें कोई ऐसा DNA का सेगमेंट सेलेक्ट करना पड़ेगा जिसका सीक्वेंस हमें नोन हो. So this is one of the variation where we can use STS sequences so that we can develop site-specific primer. If primer they are available, then we can specifically amplify the target DNA sequence. और जब हमारे पास amplified fragments आते हैं इनको purify किया जाता है and then they can be digested with a set of restriction endonucleases, or digestion के बाद we can resolve them on the gel. Because here we are going to use amplified DNA fragments and then they are digested, then they are commonly called as the caps, that is cleaved amplified polymorphic sequences are caps. Now these sequences they are very helpful even to differentiate the heterozygote. Suppose here we have three type of sequences. Let us suppose one parent having this homozygous situation A and BB, for another parent, and offspring may be AB. First is it is having two restriction side so that 3 fragments can be resolved on the gel after amplification. And then in case of another BB, here 3 restriction sites are present and we can resolve 4 DNA fragments. And in case of heterozygote like A, B, the genes of 2 parents can be represented here. For example, the dark bands indicates that they belongs to parents, like here this band and this band. It is representing one dark band in the heterozygote, and then second is here. And in this way, by using STS sequences, we can even differentiate some heterozygotes.